I'm Mike Fisk, Director of the Upstate Stem Cell cGMP facility at the University of Rochester Medical Center in Rochester, New York. I'd like to tell you about our new 3,600 square foot state-of-the-art cGMP facility for the development and manufacture of new cell therapies, funded in part by NISTEM, New York State Stem Cell Science, and the Empire State Stem Cell Board. The acronym cGMP refers to the Current Good Manufacturing Practice regulations enforced by the U.S. Food and Drug Administration. GMPs provide for systems that assure proper design, monitoring, and control of manufacturing processes and facilities. Adherence to the GMP regulations assures the identity, strength, quality, and purity of drug products by requiring that manufacturers adequately control their manufacturing operations. A GMP-compliant system of controls, when put into practice, helps to prevent instances of contamination, mix-ups, deviations, failures, and errors. This assures that drug products meet their quality standards and ultimately the safety of patients. The Upstate Stem Cell cGMP facility is a newly constructed and fully validated cGMP-compliant facility designed for the manufacture and testing of preclinical and early clinical phase cell therapy and monoclonal antibody products. Our capabilities include development of clinical scale manufacturing processes, development of analytical methods for product characterization and release, GMP manufacturing and in-process testing of clinical grade materials, as well as quality control release testing of final product. So please join me as I take you on a virtual tour of the Upstate Stem Cell facility, describing some of the main features. The Upstate Stem Cell cGMP facility was designed such that clinical trial materials manufactured and tested are in compliance with current good manufacturing practice in order to ensure product safety for human subjects in early phase clinical studies. The facility was designed and is operated as a multi-use facility with goals of eliminating product contamination, cross-contamination, and/or mix-ups that could potentially compromise the integrity of the manufactured product. The controlled manufacturing environments include HEPA-filtered air from redundant air handling units, providing clean room air quality from ISO Class 8 to ISO Class 5. Minimum pressure differentials of 0.05 inches of water column are maintained between adjacent clean rooms as well as between clean rooms and outside uncontrolled environments. In addition, room temperature and relative humidity are controlled to meet rigorously defined specifications. Procedures are in place to maximize the unidirectional flow of personnel, supplies, raw materials and reagents, products and intermediates, as well as waste. The facility was designed for optimal cleanability with the use of gel-coat polymer surfaces on walls and ceilings, epoxy-coated floors, and stainless steel work surfaces. Floors, walls, and ceilings are constructed with minimal seams and ledges. Wall mount shelving for storage is eliminated in favor of stainless steel metro shelving. In addition, equipment, work surfaces, and storage shelving are mounted on casters for easy moving and cleaning. Our facility design, operational flow, and intent of use was successfully presented to and reviewed in detail with the FDA in a Type C meeting format. The Upstate Stem Cell cGMP facility was designed and outfitted with process and analytical capabilities for the GMP manufacture and testing of early phase clinical trial materials, including stem cell therapy products and monoclonal antibody products. The controlled environment contains 3 manufacturing rooms, each with dedicated HEPA-filtered air handling, providing ISO Class 6 air quality. Each manufacturing room is equipped with cell production capability, including 2 biosafety cabinets providing an ISO Class 5 environment, 4 tri-gas cell culture incubators, tabletop centrifuge, and an inverted light microscope. In addition, one of the manufacturing rooms is outfitted with monoclonal antibody production capabilities, including a wave bioreactor with capacity up to 25-liter working volume, a microfiltration ultrafiltration system for cell separation and culture supernatant concentration and diafiltration, and a chromatography system for downstream purification. Adjacent to, on each side of the 3 manufacturing rooms, are ISO Class 7 intermediate entry and exit rooms. These areas house refrigerated, frozen, and liquid nitrogen storage capabilities for raw materials, reagents, intermediates, and final product. The Quality Control Analytical Testing Lab is a dedicated area equipped with cell culture capabilities, as well as analytical testing equipment and methods for the characterization and QC release testing of stem cell and monoclonal antibody clinical trial materials. In addition, we have access to testing capabilities at the University of Rochester Medical Center clinical laboratories, as well as the medical school core facilities that include flow cytometry, functional genomics, and proteomics. The Upstate Stem Cell cGMP facility has established and implemented a GMP-compliant quality management system, which defines a program for the systematic monitoring and evaluation of our facilities, utilities, equipment, and manufacturing processes and test methods to ensure that standards of quality are being met. Good documentation practice is an essential part of the quality system and ensures the existence of documented evidence, traceability, and records for all aspects of GMP manufacturing and testing. A key quality system element is oversight of the routine environmental monitoring program. All controlled environments are periodically monitored for total airborne particle counts as well as microbial viable counts. Clean room parameters are continuously monitored, including room temperatures, relative humidity, and pressure differentials. I hope you enjoyed the tour of the Upstate Stem Cell cGMP facility. The design and operation of the facility has been specifically developed to meet the latest in regulatory standards for the manufacture and testing for early phase clinical grade cell therapy products. The facility is currently available to academic researchers and companies wishing to develop new cell therapies, and we are currently supporting several users in their efforts to manufacture investigational cell-based products for preclinical and early phase clinical studies. If you would like more information about our capabilities, please contact us through our website.